Welcome to Synapses. I'm Gökçe Özdemir, a biomedical scientist specialized in neuroscience. And Synapses is a bridge to make medical science more understandable for everyone. Today, I have a very special guest to talk about a very special biotechnology company, FinalSpark, headquartered in Switzerland. Their main goal is to create biocomputers, wetware that combines biology with software and hardware for advanced computing. For a gentle intro, imagine a computer that isn't carved from silicon, but grown in a lab. And instead of pushing ones and zeros through rigid wires, imagine you're coaching a little choir of cells with gentle electrical cues until their rhythms can produce useful patterns. This is FinalSpark's bet. Bioprocessors grown from neural organoids that you can stimulate and read from remotely, and doing so using a fraction of the energy of today's AI. So today, with Dr. Ewelina Kurtys, who's a neuroscientist and a strategist at FinalSpark, we are asking, what can these living processors really do? How do you program them? And what are the ethics of a brain in a dish?
Hello. So my journey from biology to artificial intelligence and to FinalSpark was that I always was fascinated with the human brain. I wanted to be a scientist also. So I did a PhD in neuroscience in the Netherlands, and I was specializing in brain imaging. I was using PET, positron emission tomography, which is one of the techniques of non-invasive medical imaging. And then I also did some postdoc, but I also always wanted to go to industry, and I always wanted to discover what is outside academia. So my first job in industry was in a small company which was doing a medical imaging service, analyzing images, big quantities of images from clinical trials, and a lot of this analysis was automated. So this is how I learned about artificial intelligence, which was at that time very hot topic, especially in imaging field. And I discovered that AI actually has huge opportunities, not only in medical imaging, but I realized that there is much more. And I started to work on commercial applications of AI. And also I went a step further and started to work on the future of AI, which are biological computers we discussed today. So this is intersection of neuroscience and artificial intelligence and engineering.
Thank you for the intro, Ewelina. And before we get to the biocomputer aspect, I'm really curious about the imaging that's currently used for the brain, whether it's PET scans or functional MRIs, because there are studies where functional MRI is used alongside artificial intelligence, where a person enters a scanner, they give the person a stimulus, they look at the brain activity, and they can match the stimulus with the brain activity, keep training the AI to then try and understand what the person's thinking about just based on how their brain looks like in the scanner without needing the person to actually tell us what they're thinking about. How would this be relevant to the topic that we're about to talk about? Like, would you say that this type of imaging analysis is a bigger scale of what you're doing at FinalSpark? We're trying to understand what the brain is telling us from a whole brain picture. And then at FinalSpark, you're looking at individual cells almost that then form a community, and then you're trying to get bigger and bigger.
So what is done today with imaging is, I would have to say, a bit easier because it's just classification. So based on the activity of your brain, which can be measured non-invasively, with imaging, you can guess what a patient is thinking about, but you can classify from maybe 10 different options which you know, which you defined, but you cannot discover what person is thinking about without having any background knowledge. So just reading someone's mind. And what we try to do at FinalSpark is much more difficult because, of course, brain has some region, and we know a little bit about this. We know what does it mean when some regions are active. But what we try to do at FinalSpark is a bit more difficult because we don't have brains. We just have components, building blocks of the brain, which are neurons. And we try to understand what electrical activity, spiking of neurons means. So we don't have any brain regions. We just have neurons. At the moment, they form such a quite random structure, which consists of around 10,000 neurons each. And we try to understand the electrical activity of these neurons. And in the future, we would like to be programming them so that we want to send them some signals, chemical or electrical, and we want that they get us some kind of response which makes sense. So this will require actually translating these spikes into some specific knowledge. So far, nobody knows too much about this.
It's really a fascinating new field. When we compare to the current imaging techniques, it's like we're at opposite ends, right? The current techniques with PET and fMRI, the resolution is a huge issue, and we can't get specific enough. So we don't know the specific details. And then on the other end, with FinalSpark, it's like we know too much of the specifics, and now we have to build from here so that we can know more of the bigger picture. And you also mentioned that to do this, we use these communities of neurons, which are the brain organoids. So when we think of an organoid, these are lab-grown, pea-sized neighborhoods of cells that you get from stem cells. Nowadays, you can take a skin cell, program it back to the time when it was a stem cell. From that stem cell type, create neural stem cells or anything you would like, really, and then start creating these mini organs. And they grow, they form a neurosphere. It's a community of them. And when we like to say that they resemble a mini brain, but there is still probably many years of research until we can actually say that. This is still just 10,000 of cells in a little sphere compared to millions of neurons with their billions of synapses in a real brain. But before coming to FinalSpark, did your thoughts on organoid research change from where it was before to where it is now that you've seen its potential under FinalSpark? Because when I think of organoids, it's like, great, like we're getting closer to understanding how the brain could actually be forming naturally, how different cells could be talking to each other, forming new networks. But I feel like with FinalSpark, the organoid has to mean a whole new thing because now you're looking at it as if it's a computer.
It's funny because before I work with FinalSpark, I never actually worked with organoids. I did work with living neurons in vitro, but a bit different form, just monolayers. So yeah, I think that FinalSpark is approaching living neurons in totally different way because normally in biomedical research, we try to understand how neurons work. We try to find some cures for disease or try to understand the mechanism of human disease. But here, that's not actually the objective. Here, we try to use neuron as computational unit. So that's something totally different. It's totally different approach. And I would say that actually there is a lot of engineering and mathematics in this project. And biology here is actually quite, it forms a basis. So it's necessary to have healthy neurons. So we need to have everything correct in the lab. But the whole challenge is actually more on the engineering side because to make sense of the signals, to analyze them, to try to program neurons is actually more mathematics and engineering. So I would say here biology is just a basis, totally different approach to what normally you do in the lab with neurons.
I love that you brought me back to computation because as a biologist myself, my mind immediately goes to, okay, what are the composition of these organoids and how do you know? Because when we go from a stem cell to an organoid, it's not just neurons, right? It's a lot of different types of cells that have to come together. And I can imagine it has to be an issue that you're considering about, like, how do we keep these populations consistent across our organoids? Because a
Yes.
change in what kind of cells they would be in each organoid could, I'm sure, affect your results. So then, can you know during the experiment what your composition is, or do you have to wait until the experiment's done, you fix the cells, you stain them, and then you trace the data?
Yes, it's a very important question. You are absolutely right. And it's a big challenge. So for now, we actually can know what exactly we have inside once we kill the organoid and we take samples. And we did some mRNA analysis, which actually you can see on our website. There is section neuro platform about our laboratory. And there are also some results from RNA analysis. And we can have some idea from these results about which genes are expressed in neurons, so which kind of neurons we have, which receptors do they have. And this is really important, and it's still a challenge to make sure that everything is correct. And yes, there are different cell types also. We're still working on the on composition. Yes, but it's usually the exact biology of the cells we can have after we kill them. But what is interesting, when we are using neurons, we can actually understand quite a lot about how they work, because when we put them on the electrodes, what we usually do for our experiments, we can see immediately from the electrical activity if everything is okay, because healthy neurons, they have something like spontaneous spiking activity. So from that, you can have already some idea. And we indeed observe a lot of variability. Variability is always a challenge in biomedical research, because cells are differentiated from stem cells. So the mix is not always the same. And we also observe differences between batches. And also what is even makes the whole story more difficult is the fact that neurons are not stable systems. So they're dynamic system. That means that today your neurons are different than yesterday, or tomorrow they will be even more different. So that means that when we give some stimulation, some input to neurons in form of, for example, electrical activity, and we get some response today, tomorrow we can get another response, because it's not stable system. So apart from biological variability, we also have a challenge that neurons are not stable. So yes, there is a lot of variability in everything what we do.
And then a lot of analysis has to be done. Like, it brings me to the computational part of the engineers to figure out what each different signal means. And I bet they also have to become biologists in the process. Like, I feel like it would be impossible to actually understand the true weight of a biological signal without actually understanding the bigger meaning of that signal for the entirety of that system to work. So as good as you would be at computation, well, these are different things. I think you'd be digging a hole for yourself as a computational scientist if you're not investing time to understand the biological. And this brings me to my thoughts about a processor compared to a bioprocessor. I think we can think of a processor as just a 1 and a 0 switch, and it's easy enough to analyze. And when I say easy, of course, I'm not trying to undermine anything, but it's clear when there's a signal versus when there is not. When you come to the biological aspect, well, there are many things that can happen in a cell until we actually say it's an action potential, or we say a neurotransmitter was released, or, well, I mean, a neurotransmitter can get released, but whether it's going to cause a signal in the next cell or not, these are a lot more complex than a 1 and a 0. There's a lot of 0.25s and 0.75s. So how do we go from a processor to a bioprocessor? What is the definition of a bioprocessor for FinalSpark?
So bioprocessor is a processor which is built from living neurons. And at the moment, our prototype consists of 10,000 neurons each. And they are such a 3D structure, such a little blob of neurons, which we put on electrodes, on 8 electrodes. And you can actually see this on our website on finalspark.com. There is section live, and you can see the live view from our lab to have an idea what is this.
But then the live view, how do you actually comment on that live recording? How do you know what that live recording is actually talking about?
So from the camera, we can see the electrodes, that's dead matter, so this is sure. And on the electrodes, we put neurons. And we actually, you can also see on the website, there is also a stream of the data readout from our laboratory. So you can see activity of neurons. So we assume that if they are electrically active, that means they are alive. So there must be something. So it's of course difficult to interpret this data, but at least we know that it works, that you have a signal. And of course, this signal is biological. So there are also some challenges that you have to see the difference between the noise and the real signal, because there is always a lot of noise in biological data. But we have developed quite robust, reliable methods to distinguish what is real signals from the neurons.
And when we talk about interpreting the signals from the neurons, how do we actually signal to the neurons ourselves so that they signal back to us?
So what we do, neurons normally also in the brain, they communicate via electrical or chemical signals. So chemical signals are neurotransmitters, for example, dopamine. So we try to reproduce this in the lab. And what we do by using the 8 electrodes on which neurons are placed, we can send them electrical signals through these electrodes. So basically, we can do all this programmatically, which means on the computer, we can write a code in Python programming language. And this is all connected, of course, also to the hardware. You have a digital to analog converter, and this signal is converted to analog signal, so to stream of electricity, which can be sent to neurons. So they get electrical spike from the electrodes. So that's the input. And how we measure the output. So with the same electrodes, we can measure what is coming out from neurons. So we can also measure electrical signals as a kind of response. And this signal is also converted from the analog, because neurons are an analog. This signal is converted to digital form and in our computer can be analyzed. So basically by using Python script, you can design experiment, you can send signals, and you can also receive the answers and analyze the data.
Would you say that this protocol of electrically stimulating the neurons with dopamine and getting a signal back, like how consistently is this actually working? How would you define a failure for this stimulation to have worked, or a failure in receiving a signal from the neuron?
Well, it's actually not consistent too much because it's a dynamic system. So reproducibility is always a challenge. And well, we consider success in different ways. So one way is, of course, reproducibility. So when we do something and we get some results, then we try to reproduce this. And if it's reproduced, that means something is working. And also sometimes we do experiments with objective. So for example, we try to do closed loop experiments. So for example, we stimulate the neurons and we want them to increase the activity. And if they increase the activity, they get some dopamine released in the medium, and then we stimulate them further, and every time they increase activity they get dopamine. So this is something like kind of closed loop experiment when we have some objective. Then in this case we would say the experiment is successful if neurons are doing what we wanted. So there's two ways of saying that something was a success: reproducibility and sometimes meeting the objective which we set up front.
This sounds a lot like basic reward psychology. Like you ring a bell to the dog, you associate the bell with the treat, or I don't know, you eat a brownie and you feel good because you get dopamine and you want to have it again. Dopamine makes you want to go for that feel good event again and again. And it's like you're treating the neuron, this living being, if you will, with dopamine so that it will feel good. And it got dopamine as a reward, so it's going to want to do the same thing again and again. So the dopamine acts as your way of saying good job to the neuron. And then it's going to help it form new synapses, right? Like, would that be it? Is it more about strengthening the synaptic networks that already exist with that dopamine? Or the neuron receives the dopamine, it knows that it did a good job, and then it talks back to you saying you're welcome? Like, are we trying to strengthen what exists, or are we just trying to get the neuron to talk back?
Yes, absolutely. So actually it's both. We would like to have in vitro learning achieve, and this would mean that neurons both they can strengthen some connections, they also weaken some connections. So this is the process of learning, and this is something what we are trying to achieve, although it's very challenging. But that's indeed the objective. And this is you are right, a lot like children or animal learning, or even adults, because indeed the things which are working on the macro scale, when you look at humans or animals, how they learn by having some feedback from the environment, which can be positive or negative. So when we look at this micro level at the single cells, actually the learning has the same principles. So there is always positive or negative feedback. And by trial and error and by interaction with the environment, the neurons are learning.
This whole lab setup sounds very extremely new tech, but creating these biocomputers and training them, when I think of this, as a neuroscientist who's also worked with AI herself, I'm still struggling to get used to the idea of training individual live cells and use them as storage lockers almost. This being very, very new, what's the industry's take on biocomputers? And also, before you answer the industry's take, why should we even care about this, honestly? Like, why is a silicon processor not good enough? And why do we want to bet on this live tissue to perform better than its silicon processor alternative?
Yes, so to answer the second question, why we care. So current AI has scalability problem because the energy usage by artificial intelligence is increasing exponentially. And today we still can manage that, but we could expect that in the future it can be a problem. So we have two possible solutions for this. One is alternative energy sources and another is alternative computing. And we work on the second option because neurons are 1 million times more energy efficient than digital. So this is why we believe it can revolutionize the technology. If we can use living neurons instead of silicon, we will have very cheap, very efficient servers, efficiency in terms of energy usage, and it can be very, very competitive and can change a lot in the market. And what was the first question?
Before I remind you of the first question, how do we actually know that a biological neuron in our brain is much more efficient than an artificial neuron in an artificial neural network?
Well, actually, yes, this is a good question. Today, it's quite an estimation. It's, of course, quite difficult, but we can learn quite a lot about how neurons are working based on looking at the human brain. And there are some studies, some estimations that, for example, to simulate human brain, we would need a small nuclear plant. There are a lot of comparison which show how brain is efficient. And actually, we can see also in our everyday life that we can run very well during the whole day on banana, while ChatGPT needs millions every day for energy consumption.
Now that you answered it, that sounds really common sense. Like, yeah, obviously, the way we function every day, the way our brain does everything so effortlessly, it has to be very efficient compared to when we use AI. And, well, it uses a lot of energy to a point where it's creating even environmental concerns about the future of the power that we're going to require to use these systems at even bigger capacities than we do now. So wouldn't it be so much nicer if we could just rely on our own brain's very efficient energy handling and its own ability to learn and remember to store that information and keep recalling it? And this is not to undervalue AI. I think AI can be the wind beneath our wings. And I think it can be difficult for people who are going to try to catch up with it. But for those who can actually create that wind beneath their wings using the AI, I think it'll be fascinating. Combined with the power of our own brain, there's a lot of potential to explore there, as long as, we don't forget that our own brain's capable of great many things. And now to go back to the first question I asked you, what does the rest of the biotech sector feel about this? Like, where is this sector going for bioprocessors and wetware?
Yes, so actually this is totally new industry we are creating and there are still not many players. Our main competitor is Cortical Labs. They are based in Australia and they also want to do a biocomputer. They have a little bit different approach than us because they want to build, they actually did already the prototype of a portable computer, while we believe more in bioserver, which would be central computer. But of course, we both aim for the same. And we are very happy actually we have competitors because that means the idea maybe makes sense, not only for us, also for other people. And actually there were some research since maybe 30 years. One of our scientific advisor, Steve Potter. He was a professor. He did a lot of research, which are kind of corresponding with what we do. But there was always, of course, challenge. Nobody built such a robust laboratory like we have, where you can do automatic experiment on neurons. And they now can live around 3 months, which is very big success in the industry because normally they die very quickly. So there were some attempts, but there were a lot of technical barriers. For example, how to get neurons, because 20 years ago it was very difficult. You had to basically sacrifice animals to get some neurons from the heads. And today we have new methods, get neurons by differentiating human skin So that actually make it possible to do such a research. This is new field which maybe exists since 10, 15 years.
I want to emphasize what you just said about how long you can keep a brain organoid in the lab alive. When the field first started, keeping an organoid alive for just a couple hours would have been a miracle. And now you're talking about 100 days of an organoid staying live in the incubator. And when we say live, we're talking about it's still the clump of cells. It's not an organism. We're not talking about a consciousness, but it's the clump that has to survive in that biological temperature in the incubator. And the cells are still healthy, they're firing action potentials, there's active networks, there's active electricity in that. And when we say the death of an organoid, we're talking about how these elements, these cells start separating from this sphere because they're dying off. There is no longer any active electrical communication and no new functions that are happening. And now with the 100 days, you're saying it's become standard to be able to keep them alive for this long. When the organoid field first started, and even today, researchers still keep multiple incubators because if in case one incubator gets a contamination, which can happen quite often, your organoid that's been alive for months maybe can just die because of a contamination. And unless you have spare organoids and spare incubators, you might have to wait several months again until you can continue your experiment. So 100 days being standard protocols, it's a great success. Now, the follow-up question to that is, how do you actually define real maturity of this organoid? How much of a maturation of the tissue do you see in 100 days? Like, is there a point where it reaches a certain maturity and then it stops, or does it stay constant? Does it keep on maturing? How do you define that?
Well, this is interesting question. So when we make neurospheres, we assume they are mature already. So we assume that we put on the electrode mature neurospheres. However, indeed, they are changing over time. And this can be seen in the results of the RNA analysis, which we have done. So this is also a challenge for us.
And by maturation, I mean, yes, like these are mature neurons, but as time passes, I can imagine new synapses forming. A neuron that didn't used to have a connection with a neuron elsewhere in the sphere can form a new synapse as more time passes. So, and this may or may not be true, I'm not sure, but I just wonder that about when you analyze the data, I'm guessing you would have to normalize based on the age of the organoid and the maturity correlated with that age. Or is that maybe part of the future considerations you would have? I'm just wondering, how do you actually normalize across these organoids when it comes to their QC metrics or their transcriptomics, their firing stats? How can you compare results, whether in your lab or with labs elsewhere who might be using this technology?
Well, for the moment, nobody has the same lab as we do. Yes, you are right. It's a really big challenge, I think, the normalization. At this stage, it maybe doesn't matter so much, but we would for sure need to correct for that in the future. And yes, we hope that neurons are forming new connections. We really hope so, because we try to stimulate them all the time. Although it's very difficult to measure, actually, you cannot really measure what is inside. There are some mathematical methods to get some ideas about connectivity inside the neurosphere, but it's very difficult. So yes, we cannot check this very well, but we are hoping that the neurospheres are changing. They are forming new connections, and maybe in the future, absolutely. And also for scientific papers, because some of our collaborators who are using our lab remotely, they publish also scientific papers. So I think for that, it's also necessary to correct for the time, for the age of the neurosphere, to make them consistent.
It's so strange. The science we do, even the science that we start from scratch, from one cell, so that we can understand what happens between the one cell and the bigger brain, even then we get lost somewhere in there and we start saying, oh, things that happen in the background. We don't exactly know what happens in the background. Ever since the start of neuroscience, we would love to say that we know a lot about the brain, that we know every single thing that happens in every single part and each of the neuronal communities that exist in each part. When AI first came up, like people said AI is going to be better than the human brain. And most of neuroscientists were like, well, you guys, we don't even actually know everything about the human brain. So how can we mimic the brain with AI if we don't even know the brain itself yet? And another thing that we still don't know about the brain brings us more to the ethical boundaries, because we don't really know what consciousness really is. Before maybe asking you to define consciousness, which will be a difficult one, I'm going to ask, currently, or do you envision there to be any scenarios where a task or a stimulation would be off limits for your company just based on ethical grounds?
I think it's difficult because, well, I don't think that this blob of neurons can feel pain. However, well, at this stage of the development, we just have little 10,000 neurons. One day, when we make intelligent machine, using living neurons, we cannot exclude this option. And that's why actually we get in touch with philosophers to help us to think about this, because of course we are not competent to talk about consciousness and philosophy, but we try to encourage philosophers to think about this, if there are any limits, if there is any consciousness, because of course, if you want to assume that some of the signals, some of the simulation are off limit, that would mean that they feel pain or they are conscious. So these are the topics definitely to philosophers for the future. And we want to be prepared. That's very important for us. And also we want to be socially acceptable. So that's why we encourage philosophers to think about these options, ethical considerations, and that they help us to actually answer this kind of questions. If there is any limit, if there is something we shouldn't do, how we should think about these living computers in the future.
I think you have to be the most open-minded AI neuroscience researcher out there when you're openly saying you're asking for feedback, asking people to tell you what to consider. Because, I mean, honestly, best science happens when people collaborate. And I know that we want to kick this to the philosophers' study desks, but I will push you one more time. And I will ask, in your scientist's opinion, when will this living computer be considered conscious? And for your sake, I'll try to make it more scientific sounding and say, what would be your best argument that current organoids lack the complexity for subjective experience? And what would be your scientist's tripwire for pausing if evidence changes?
This is very difficult question. I think I am definitely not an expert. I personally think that at the moment, if you have, cells in vitro, normally you don't, of course, expect them to be conscious because they cannot process any information. And at this stage, we have stored 1 bit of information. So this is really little. And I also believe personally that consciousness is connected to our bodies. So that's not brain alone. There is a lot of evidence for this in neuroscience that actually our feelings, emotion, especially this part, not necessarily the rational, but the feelings, emotion is strictly connected to how our body works. But of course, we cannot exclude consciousness of living computers. I think that it's the same problem as with digital, because there is also a lot of discussion. There are some people who believe even that today artificial intelligence is intelligent. So I think when there is processing of advanced algorithms, there is always a doubt of is it conscious. So I think the time when we'll be able to process advanced information or have some conversation with the biocomputer, that's the time when we can ask about consciousness.
I have to say that was a really good answer. Current AI, the bioprocessors and wetware aside, I don't think AI can be conscious at all. I mean, I think about how a processor works and it's 1 and a 0, each part is not part of the whole. The one part doesn't know about the whole. Whereas you look at the human, how the human cells operate, and you see that each part carries the entire information of the rest of the body. I mean, even consider the organoids that you build from stem cells. Each cell in your whole body carries that same genetic information. It just uses that information differently to do different functions, which is different than how a computer works. And biology is not just ones and zeros. But when we come to the point where we can actually create bioprocessors and biocomputers, like you said, start communicating with them, then I'll be like, "Oh, wait, okay, it turns into one and zero eventually to give us the signal, but it starts off as something else." At that point, I think we'll be having entirely new and different conversations. And I don't think that we can even predict what these conversations will entail.
Yes, absolutely. It's hard to predict. But actually, we would like to run the same things which are run on digital. So we imagine that generative AI will be possible both on digital computer or biological computer, but with lower energy consumption.
What's actually needed for the future of FinalSpark and for these bioprocessors to improve to the next level? Let's say you could double the budget you had tomorrow. What's the bottleneck that you would want to attack? Is it something about your sensors or your neural protocols, your automation, or maybe just hiring a philosopher to figure out this consciousness thing out?
Actually, philosophers are not needed on our team because we prefer that someone is doing research externally, independent of us. I think it's much more reliable. But what is a bottleneck? So it's very expensive actually when it comes to people because you need a very specialized team to work on biocomputing. It has to be people who combine both engineering and biology, as you mentioned before. So that's the bottleneck. That's why we talk to investors, because the project is so far self-funded and we are now considering external investment. We are seeking 50 million Swiss francs, which can accelerate significantly our development. And of course, this money would be mostly spent on people. Of course, we need also a lot of hardware improvement, but then the biggest cost there is also people, because like the pieces of hardware is not the biggest price, but more the people who have to work on that. So we have a lot of ideas on how to improve, how to increase the number of electrodes, how to improve all what we have in our laboratory. And of course, to run a lot of experiments. We need to run a lot. We need to analyze the data. So there is a lot of work to be done. And that's mainly the people are needed. And our biggest now, the next milestone, which we have at the moment, is learning in vitro. So far we managed to store 1 bit of information. And with investors, in the time of 2-3 years, we want to be able to crack basic learning. So basically we want to change the connections between neurons for basic tasks. So that's the future, and that's the biggest bottleneck what we need.
When you say change neurons for basic tasks, let me confirm that I got that right. We're talking about learning to command them. In response to that command, we want them to not only change their chemical signal, but actually start changing morphologically. It's a biological system that's not only chemically changed, but also physically changed. Is that right?
Yes, absolutely. Physically, because we would expect that neurons will have different connections because that's learning. And we will measure this most probably by, like we do today, by measuring the changes in electrical activity of neurons. So these changes in electrical activity, they reflect the changes in connections between neurons.
And you also mentioned 50 million Swiss francs to accelerate this and to go from that 1 bit of storage to cracking learning and letting neurons change themselves completely. Say you're an investor with maybe not the whole 50, but even if just the 1 million, if you were the investor with that money, do you think there's anything we haven't yet touched on that you would want them to hear? Is the most essential thing for an investor to get from this episode?
I think for investor, the most important is always the money. So we actually expect that this will be very profitable technology because, as I said, if we can provide computational power, which will be 1 million times less, using less energy, 1 million times less energy, it will be very, very competitive. So we basically can run generative AI much, much cheaper, which can make a huge difference. And we actually made some projections that in 2033 or 36, we will make billions of profit, not even revenue, profit. So it's expected to be extremely profitable. I think that's a good argument for investor. But of course, the challenge here is, which also should be considered, that it's also long-term investment. And it's also high risk investment because deep tech investments are always risky. It's not possible to guarantee the timeline. We cannot predict everything, what will come on our way. So there is always a risk involved. So of course, this is not investment for someone who would put the only money they have in such a company, but rather a small fraction because it's high risk investment. And of course, we prefer to talk with institutional investors, with people with venture funds. It's a bit more difficult if private people would like to invest. Although we get a huge amount of questions from private people who would like to put their own private money. And we are also considering accepting this kind of investment from private people. Although it's always very important to emphasize that it's long-term and high-risk investment.
I really applaud you for highlighting that over and over because I think it's not just tech investment, it's a biotech investment, right? You need both the tech and the bio to work both individually and also coherently. And anything bio, we know it can take years and years, and the reward can be very high, but it's not a guarantee. And a lot of years can go by until you actually know the value that your risk's going to bring or not. And everything you're saying about the potential of these systems, and I'm really excited for years to come and where these topics will inevitably change and we'll see where we actually go from here. And the rest of the industry will maybe take a pause and ask themselves if they should be reconsidering their approaches. Just think about how you're using multi-electrode arrays to record from your organoids to then create an intelligent system when these arrays are already being used today for different purposes, right? Like they're solely used to record from communities of neurons, record their communication and their synaptic activities. And I think companies like you and future companies to come, there's great potential for new questions to be asked, not only about how existing technologies can be used for different purposes, but also about how new tech can be developed to ask new, far and advanced questions. Okay, Ewelina, thanks so much for this chat. I really, really enjoyed it so much, and I absolutely cannot wait for you to receive the funding that you guys so well deserve and for us to see what this funding will bring to the future of wetware and bioprocessors and biocomputers. Thanks so much for your time, and thanks everyone for tuning in. If you have any philosophical or scientific questions, please make sure to let us know. Thanks. Bye.